following are the 5 common types of functional brain imaging modalities used in the cognitive neurosciences and related field to study how human brain systems work and are connected, which are magnetoencephalography, electroencephalography, positron emission tomography, functional magnetic resonance imaging, and functional near-infrared spectroscopy. I will briefly describe these imaging modalities after this. Before describing functional brain imaging modalities, I would like to show the trends of publication related to functional imaging modalities in the cognitive field. I use PubMed database to explore or identify relevant studies by using cognitive and fMRI as the keywords, and the second one is cognitive and EEG as the keyword, followed by cognitive and MEG as the keyword, then proceed with cognitive and NIRS as the keyword, and finally cognitive and PET as the keywords. PubMed database shows an increased patterns of articles published until August 2020. Based on the number of articles published, functional magnetic resonance imaging and electroencephalography show a promising neuroimaging method in cognitive studies as compared to other functional imaging modalities. Functional MRI and EEG indicated as the well-described and familiar neuroimaging methods on the literature to everyone in the field. Although functional near-infrared spectroscopy is still a new method in functional brain studies, it indicated an increasing pattern in publication. The different number of articles published in different functional imaging modalities is shown by PubMed database might be due to various advantages and limitations for each of these modalities that will be discussed after this. From this trend, what we can summarize is the increase in publications each year in functional brain imaging modalities indicate the development and importance of these neuroimaging modalities in the exploring brain, mind, and behavior. The research covers many areas, not only focus on the clinical implementation, but also leads to knowledge improvement, inclusive advancements of health and wellness in many ways. Nowadays, people try to protecting and improving their health, focusing on keeping people well rather than treating or managing illness or disease. Now, I hope you are ready to understand the functional brain imaging modalities briefly. Next, I will give you an overview of functional magnetic resonance imaging. In the early 1990s, a physicist named Seiji Ogawa discovered functional magnetic resonance imaging. He found that molecules in the blood that are oxygen-rich or oxygenated have different magnetic properties than those that were oxygen-poor or deoxygenated, and that this difference in magnetic properties results in a small change in the local MR signal. In 1991, Jack Bolivier had performed an fMRI study using external contrast agent called gadolinium. In this study, Bolivier generates the first fMRI maps of human task-based activation using a visual stimulus paradigm. In 1992, there were 3 groups from different laboratories, which are Bendettini et al., Kong et al., and Ogawa et al., simultaneously and independently published first functional images using the Bolt mechanism without exogenous and contrast agent. Therefore, Ogawa used blood oxygen level dependent or bold contrast to map brain activity. How does fMRI work? fMRI expands the use of MRI to measure changes in the magnetic properties of blood in the brain. In an active state, for example during a visual task, motor task, or cognitive task, neurons of activated brain region demand more oxygen than they do in the normal state or without tasks to process information. Oxygen consumption increases with the demand of the task. This changes the magnetic properties of oxygenated and deoxygenated in the blood vessels within the activated brain regions, which induces magnetic susceptibility differences between the blood and the surrounding tissues to obtain MR signals in the brain that allow the study of brain function. This results in an endogenous contrast agent called blood oxygen level dependent or BOLT in fMRI. Blood oxygen level dependent contrast is the most commonly used contrast mechanism to map brain activity. As for benefits, functional MRI is a safe and non-invasive method to examine human brain function. However, as for risk, fMRI follows the same risk like MRI. Next, I'll give you an overview of electroencephalography or EEG. EEG was discovered in 1929 by the German psychiatrist Hans Berger. The development of EEG proceeded in the certain phases. In the early days, with first measurement of weak electric processes with sensitive voltmeter. At this phase, researchers aimed to interpret the observed oscillatory variation. There were three main researchers in the early stage, which are Berger in Germany, Adrian in England, and Gray in the US. The second phase was aimed to improve multi-channel recording techniques on paper, like in typewriters. Start from here, the development of EEG significantly progressed, which is progress in technology included the electronics and the application. The interpretation of observed rhythm of the EEG became a tool in medical practice and research. EEG provided considerable insight into our understanding and clarification of electrical activity or brainwave of the brain. How does EEG work? The brain contains billions of nerve cells, also called neurons. These neurons produce very small or tiny electrical signals that form patterns called brainwaves. During an EEG, small electrodes are placed onto the scalp of a patient or participant. The electrodes detect the brainwaves, then the EEG machine amplifies the signals and records the wave pattern in a computer with designated software for further processing. A diagnosis or clarification of the brain's electrical activity, either regular or abnormal, depends on the pattern of the brainwave. The brainwave or brain oscillation can be distinguished by several frequency bands, which are delta, theta, alpha, beta, and gamma. As for benefits, EEG is a safe and non-invasive method to examine the electrical activity of the brain. EEG has been shown to be a cost-effective or cheap tool to explore human brain function. As for risk, the spatial resolution is poor as compared to fMRI method. A potential or possible complication affect patients with seizure disorders, as the flashing light there are parts of the test may trigger a seizure attack. Next, I will give you an overview of magnetoencephalography or MEG. History of MEG started in 1963, whereby Gerhard Bohler and Richard McPhee from the Department of Electrical Engineering, Syracuse University, New York, had recorded biomagnetic field from the human heart. They used two identical coils connected in series as detector. These coils were placed parallel to each other on the subject chest. The recorded data was very noisy. As we can see from this figure, the biomagnetic fields are very small compared to urban noise. However, the finding from Bohler and McPhee was still valid. In 1968, David Cohen, a physicist from the University of Illinois, measured first imaging signal using copper induction coil as detector. As we knew from the findings from Bohler and McPhee, noise becomes a challenge in MEG measurement. Therefore, David Cohen done the measurement made in a magnetic shielded room to reduce the magnetic background noise and to improve the sensitivity of the recording. Later, in 1969, David Cohen measured MEG signals using the superconducting quantum interference device or SQUIDs that just had developed by James Zimmerman as a detector. The recorded signals were clearer than before and almost as clear as those of EEG. Start from here, the development of MEG keeps improving, for example from a single SQUID detector until multiple sensors were introduced. Recently, spin exchange relaxation free or SURF magnetometers and chip scale atomic magnetometers were introduced. The application of MEG in neuroimaging and human cognition exploded in the 1990s. How does MEG work? MEG measures the magnetic fields produced by electrical currents in the brain. The neuromagnetic signals generated by the brain are extremely small than the Earth's magnetic field strength. Therefore, SQUIDs or SURFs or CISM sensors were used to detect and amplify the magnetic fields generated by the neurons. As for benefits, MEG is a safe and non-invasive method to examine the magnetic field's activity of the brain. The temporal resolution is high, which is around millisecond as compared to functional magnetic resonance imaging. However, for risk or the limitation of the MEG, the spatial resolution is poor, which is within centimeters, MEG has shown to be an expensive tool to explore human brain function, as an extra budget around more than 5,000 ringgit Malaysia needed every week just to pay for the helium of squid sensors to reduce the temperature and produce low impedance in order to detect and amplify magnetic fields of the neurons. Next, I'll give you an overview of positron emission tomography, or PET. In the early 1950s, David Cole and Roy Edwards were introduced the concept of emission and transmission tomography. In the early 1950s, Gordon Brownell and George Sweet developed a different technique for radionuclide scanning to enhance spatial resolution. In 1951, William Sweet reported the first medical application of the positron annihilation radiation to localize brain tumors at Massachusetts General Hospital. In the same year, Rand, Good and Handler published studies of positron annihilation to localize brain tumors. These concepts led to the innovation and development of PET technology in the clinical and research setting. In 1974, Mike Phelps and Ed Hoffman developed the first human PET. Step from here, the development of modern PET scanners keep expanding. How does PET work? Positron emission tomography is a technique that observes and measures physiological function by looking at blood flow, metabolism, neurotransmitters, and radio-labeled drugs. PET uses a radioactive substance called a tracer to look for disease or injury in the brain. PET works by using scanning device or a machine with a large hole at its center to detect positron emitted by a radionuclide in the organ or tissue being examined. PET allows the measurement the relative changes over time of a disease process evolve or in response to a specific stimulus. PET shows how the brain and its tissues are working. As for benefit, PET scan can measure the changes in cellular level metabolic take place in an organ or tissue, which gives an advantage over other technologies in discovering lesions in the body, either benign or malignant. As for risk, although a radioactive tracer is injected into the patient during a PET scan, the amount of radioactive drug is small and short-lived. The small amount of radioactive tracer is not enough to affect the patient. However, there are risks due to the tracer, which are a potential reaction or an allergic reaction. Expose your unborn baby to radiation if you are pregnant, and expose your child to radiation if you are breastfeeding. Next, I will give you an overview of functional near-infrared spectroscopy. In 1977, Franz Dobsis from the Department of Physiology of Duke University discovered that oxygen contained in the blood gives off light wave close to infrared range. First near-infrared spectroscopy clinical studies was in 1985 on newborns and adult cerebrovascular patient to study cerebral oxygenation. Start from here, near-infrared spectroscopy keep developing. In 1992, first functional near-infrared spectroscopy studies using single-channel instrument was carried out independently by Chance, Kato, Hoshi, and Wieringer. Functional NIRS is use of near-infrared spectroscopy for the purpose of functional neuroimaging. FNIRS technology is still young, more study needed to improve this technique. How does functional near-infrared spectroscopy work? Functional NIRS is an optimal imaging method that uses near-infrared light, approximately 650 up to 1000 nanometer, to measure concentration changes of oxygenated and deoxygenated hemoglobin in the cortical brain structure. Functional NIRS principle is almost as fMRI method, whereby it measures the hemodynamic response or blood oxygen level dependent response. In principle, light from the near-infrared range has the ability to penetrate biological tissue, for example, skin, skull, and brain. The light waves is mainly observed by oxygenated and deoxygenated hemoglobin. Therefore, by using special light detector, this enables to collect and measure the amount of light being produced by the changes of the cortical oxygenation in the blood. As for benefits, functional NIRS is a safe and non-invasive method to measure the human brain activity. Functional NIRS relatively high temporal resolution, which is around millisecond. Functional NIRS recording can be done in natural body position. Functional NIRS is low sensitivity to motion artifacts than fMRI. Other than that, functional NIRS has been shown to be a cost-effective or inexpensive tool and also portable to explore human brain function. As for limitation, functional NIRS relatively low spatial resolution, which is within centimeters. Other than that, it can only cover cortical brain. Functional NIRS measurement also influences of extracerebral hemodynamics and hair and skull characteristics. This figure shows the spatial and temporal resolution profile of the most used brain imaging modalities. Temporal resolution is depicted on the horizontal axis, and spatial resolution is aligned on the vertical axis, both on a logarithmic scale, with indications of relevant periods of time and brain structure. At the moment, there is no most effective neuroimaging modality. This is because of each modality have a different temporal and spatial resolution advantages and limitation.